Good evening. Kieran is a third-year PhD student in analytical chemistry at the University of Alberta. In 2019, they completed their Bachelor of Science in Biochemistry at the University of Waterloo. Before joining the Harynuk group, Kieran is working in the development of sample preparation methods for metabolomic analyses of biological samples. Welcome, Kieran, and thank you very much for coming to present to us.
So thank you for that introduction, Paula. Today, I will be talking to you guys about just a bit of the roles of scientists in society, as well as the impact of metabolomics research. So I am a student at the University of Alberta, which is part of the Metabolomics Innovation Center. This is Canada's national platform for metabolomics research, training, and services. And what we do is we measure small molecule metabolites in biological samples or just any samples that have metabolites in them. And there are a variety of tools and applications. So TMIC is made up of 5 universities with 8 different nodes or research groups. And in particular, I am part of the Harynuk node, where we do a lot of untargeted analysis, which basically means that we're looking for anything and everything that we can find. We don't have anything like specific that we're looking for. We deal with volatiles, which are compounds that can be made into gases. And we use a very powerful instrumentation technique known as comprehensive two-dimensional gas chromatography coupled to time-of-flight mass spectrometry. And I will be explaining that a bit later on in the presentation. So to start off, I'm just going to go over some definitions really quick, because I'm sure you guys know these. So science is basically a system of knowledge that is about the general truths of the physical world, and these are obtained through the scientific method and through experimentation. A scientist is a person who specializes in a particular field of science, and they do scientific work or experiments. And then society is a community or a group of people who have common traditions and collective activities and interests. So as I mentioned, I will be mentioning what the role of science in society is. And the role of it is essentially to change society. The way that this is done, or science has contributed by providing us with longer, healthier lives, ability to monitor our health, and also having medicines and cures for diseases. It provides us with food and water, and it also provides us with communication technology. So I am able to do this presentation from my home, and you can watch it from anywhere in the world. The challenges are that we need to ensure that governments and citizens are scientifically literate. So that's why I really like that there's this science outreach, being able to share scientific knowledge with other people. So the definition that I just showed is from UNESCO, and this is Science for Society. Now, going over a little bit of history of science, I'm not going to go over all of these scientific contributions, but I am just going to list a few notable ones that people will know about. So in 1666 was when Isaac Newton theorized gravity when an apple fell from a tree. We have Charles Darwin's evolution of the species and natural selection. Later on, we had the discovery of insulin, which is a hormone that helps us to break down sugars and has helped people who have diabetes lead normal lives. So it saved lots of lives. The discovery of penicillin, which is an antibiotic. This sparked research into other antibiotic drugs so we can treat infections nowadays. The discovery of the DNA helix started a lot further research in biological sciences. The first test tube baby, and this technology is still used today for in vitro fertilization, which helps people who are struggling with infertility. Another one people might be familiar with is Dolly, who was the first clone from an adult mammalian cell. The completion of the Human Genome Project. So they obtained the entire sequence of the human genome and all of the genes. and as well Google, which allows us to have information, any information that we want right at our fingertips. So again, I'll reiterate what the role of science is to change society, but is it always to make society better? Well, it hasn't always been used for the good of society. Science has led to the development of nuclear weapons. There's been lead used in paints. It helped it to dry faster, but we now know that lead is dangerous. And this was used for common household items, as well as pesticides used to reduce pests that are destroying crops, but has also been bad for our ecosystems. And these kinds of things can cause people to lose their trust in science, which leads into misinformation, which is when incorrect or misleading information is being shared and stated as a fact. And this can be done either intentionally by someone knowingly sharing false information, or by someone reading a misleading article, thinking that it's true because it's published online or in a journal, and then they share that information and rumors can begin. And as scientists, we're often having to fight back against misinformation and demonstrate the truth through our research. And something that has helped to increase people's trust in science is understanding how the scientific process works. And the way this is done is as scientists, we present our findings in a variety of ways. We come up with conclusions from the results that we obtain. But the world around us is always changing. We're always learning new things. And we always want to share that knowledge. And it's important to be honest about when we are wrong, because ultimately our goal is just to share the truth and to learn. So for example, in this infographic back in the 50s, they were saying that you should use butter because it's healthy for you. And then in the 1980s, they were saying, no, you should actually use margarine, it's healthier. But recently in 2020, they discovered that neither or is like healthier for you, that you should just choose it based on your health needs and discussions with your doctors. And regardless of this, misinformation is still all around us. An example of misinformation are the claims that are made by a food blogger known as Food Babe, and she uses pseudoscience and misinformation to scare people about the food that they eat. And some of the claims that she has made are that there's just no acceptable level of chemicals that you should ever ingest, that sciencey sounding ingredients can be deadly toxins, as well as that if a third grader can't pronounce an ingredient, you shouldn't eat it. Now here I have an ingredient list of a food item, and going by Food Babe's logic, I would not want to eat this. There are a lot of chemicals in this. There are words here that sound very sciencey, and I wouldn't expect a third grader to be able to pronounce all of the words in here. But you'd be surprised if I told you that this is an ingredient list to natural pineapples. And I love pineapples, so I'd be very sad not being able to eat them. And in this ingredient list, we see things kind of like sugars, amino acids, fatty acids. These are all examples of metabolites or small molecules, which lead me into introducing the metabolome, which is a complete collection of small molecules that we find in biological and environmental samples. And these can either be endogenous, meaning that they come from inside of an organism, exogenous, so they originate from outside or from the environment. They could be toxins, and they can be indications of stressors or exposures to some chemicals. Now, according to the Chemical Abstract Services, as humans were exposed to harmful substances on a daily basis, there are millions of chemicals that are already registered as being harmful and thousands being added to that registry every day. And these can be from anything in our environment, from, for example, volatile compounds from our carpets, if you use gasoline, any pesticides, for example, if a neighbor sprays pesticides to prevent weeds from growing on their lawn, medications, so it's always really important to take medications under the supervision of a doctor, any cleaning agents, as well as any smoke or exhaust products. Now, to understand metabolomics, I'm going to be explaining a little bit about the omics timescales. So omics is just a study of different things. So for example, you may be familiar with genomics, which is the study of the genome. And in general, genomics is pretty static over time. The genes that I had when I was born are the same as the genes that I have now, or the same as the genes that I'm going to have 50 years down the line. Another study or another omics technique is proteomics, which is the study of proteins. And with proteins, there are changes, but they're not very drastic and they're not always immediate. So as you can see, for example, if you're starting out a workout routine or you're consuming more protein, you're going to have a lot more protein in your body, or your body is going to be producing more proteins, and we can see those changes. Now, metabolomics is a lot more complicated. There are many small molecules, and there are many changes. These changes can either be like really rapid, for example, this neon green line, or they can be pretty slow over time, like this blue line here. And in general, metabolomics is like a chemical detective. We can learn a lot about an organism based on the metabolite changes, and as well these changes, because they happen so fast. We always have to find ways to measure the metabolites in samples and come up with conclusions from that. So that leads me into how we measure the exposome, which is our exposures, exposure chemicals, as well as the metabolome, the small molecules. And just in summary, the way we do this, we collect some samples, we bring them to the lab, do some chemical analysis. From this, we obtain some data. And by analyzing this data, we can then come up with some conclusions. And one of the techniques that we use, and the one that we use mainly in my lab, is known as gas chromatography, which is a technique that's used to separate a mixture of compounds. Now, with this, we start off with using a carrier gas. For example, in many cases, we use helium because it's a gas that doesn't interact that much with compounds. And what this does is it moves the compounds through the column. So then we move on into the inlet, which is where we inject our sample. And this inlet is heated, so the sample instantly is vaporized. The compounds in the inlet then enter the column, being carried by the carrier gas, and this column is found inside of an oven. And we can start off with a very low temperature and slowly increase the temperature. And this column is where the separation of those compounds occurs. So the column is coated on the inside with the chemical that interacts with those compounds. And the way the separation happens, for example, we start off with the mixture. Some compounds don't interact that much with the column, so they move through the column faster, whereas other chemicals have a stronger attraction for the chemistry of the column, so they will stick around a bit longer, and we obtain separation. After this, our compounds enter the detector, and there's many different types of detectors, which I will not really get into today just for the sake of time. But what the detector does, it produces a signal which we can measure, and then we obtain our data, we can make conclusions from this. However, for metabolomics samples, this isn't always sufficient. For example, in here, we can see a chromatogram. So the chromatogram shows the retention time, or the time that the compound spends from the moment it's injected until it reaches the detector. It's on the x-axis, and then intensity on the y-axis. And we can see a lot of these little lines. Each of these lines is a peak, so it's an individual compound. And we can see that there are a lot of peaks. A lot of them are very close together. For example, over here, there's just a big blob. Same with over here, it's a very thick peak. So if there was a compound that I was looking for that came out at that time, it would be really difficult for me to say or to separate it using this technique. So how do we improve this? Well, we use comprehensive two-dimensional gas chromatography, or GCGC. And the way this works, so similar to the previous slide, we have our instrument, but instead of the column being connected to the detector, it's instead connected to a second column that has a different chemistry. So it separates the compounds based on a different property. And then after that, we go into the detector. And in the end, we obtain something that, if you look at it in the one dimension, looks a lot like what we see up here. But if we rotate it out, we can see a lot more. So in this case, on the x-axis is the time that the compounds spend in the first column, and the y-axis is the time in the second column. And then the colors indicate the intensity. So red is more intense, whereas the lighter like green and light blue colors are less intense. And here we see many, many peaks. And in particular, here, this is a result of the reagent that we use in preparing the samples. And we can see that they actually hide a lot of the compounds, for example, some amino acids or fatty acids, which we wouldn't be able to see if we only used the one-dimensional gas chromatography. Now, some applications of GCGC include, well, basically anything. We can analyze basically anything and everything as long as we can make it into a gas. So it's a great platform for non-target discovery, meaning that we're just looking for anything that we can see. And in our lab, we use it for studying biofluids, for example, urine, biosolids like feces. We can study the different components of plants, and same with the volatile emissions. So for example, if you've ever smelled a rose, we can study the compounds that are being emitted, or the smelly compounds from plants. We've also analyzed food and beverages, cannabis samples, fabrics, especially like workout fabrics. They can collect some sweat, which again goes back to biofluids. And we can also use it for environmental analysis, so studying water and air. Now, because we have so many different applications, we need to find ways to introduce these samples into the instrument. And there's many different ways, and the type of injection method just depends on what exactly the question that we're looking to answer. So we could inject straight liquid into the instrument. We can analyze the headspace, so the air that's right above the sample. We can use solid phase microextraction, which I will explain. These three I will explain a little bit later on. Thermal desorption, so using heat. Dynamic headspace, which is similar to the direct headspace, except we collect a lot more. And then cool injection in the case that you have compounds that are very volatile that will go through the instrument really fast. You want to inject it while it's cold, so it kind of traps them for a bit longer. Now, with our instrument, we have a multi-purpose sample automation. So we have a robot that does all of this because it would just take forever for us to do all of these analyses by hand. And as I mentioned, we use solid phase microextraction, or SPME. And what this is, it's kind of like a syringe, but instead of it drawing in liquid, it has a fiber. And the fiber is coated with a chemical that adsorbs or absorbs a molecule, so it acts kind of like a sponge. And you can either put it directly into your sample or put it into the headspace, or the air right above the sample, and it will collect compounds in the sample. The other technique that we use is the dynamic headspace, or DHS. And for this, we have a tube that is packed with an adsorbent material. So again, that chemical compound that absorbs and acts kind of like a sponge. We connect this above our sample. So in our sample, we heat it so that, again, like for example, in here, the compounds go into the air above it. And then we flush gas through and into the tube so that the compounds get trapped on this adsorbent material. Afterwards, we dry it down because we don't want any water. And then we use a thermal desorption. So heat, essentially, to heat this tube, and the compounds get released from that adsorbent material and then enter the column and are able to be separated. Now, how do we decide which method to use? So in this case, we were analyzing blueberry samples, and we wanted to know what the flavor compounds of blueberries are, what makes blueberries taste so good. So we analyzed them with SPME, and we can see a lot of peaks. We see over 1,400 peaks, and we see many different types of metabolites. And then we also used dynamic headspace for these samples, and we see over 4,000 peaks. So if I asked you which of these techniques is better, a lot of people would say that maybe DHS because we see a lot more, but that's not always the case. For example, in DHS, if I was looking for very volatile compounds, which come out really early from the column, DHS wouldn't be the most ideal in this situation because we have a lot of smearing, we don't have very good separation, whereas with SPME, we do have a lot of that separation. Whereas if I was looking for more of these compounds here, the ones that we see the bubbles, those ones are more intense with the dynamic headspace, so we would then choose the dynamic headspace. Now I'll go over some other applications of the GCGC instrumentation at our lab. And I will start off with environmental exposures for exposomics. So to start off, I'll go over the air story. We can analyze air, water, soil. And if I wanted to study air, well, how are we going to collect the sample? I can't just grab a bag, put some air in it, and then bring it to the lab because I don't know the volume of the bag, and I could easily lose compounds from the bag. So in our lab, we actually have a way to analyze air. And the way we do this, we have a tube, again, that is packed with adsorbent material. And an example is activated carbon, which a lot of people use just to eliminate odors in different parts of their home. So this sorbent material absorbs molecules from the air, and we connect this tube to an air pump that has a flow of about 50 milliliters per minute. And then we put it outside. So there are molecules in the air and they get drawn into this tube, they get trapped. Afterwards, we cap it, we bring it back to the lab and put it in the thermal desorption unit. So what that does, it heats the tube that has our compounds and flushes gas through it as well. Those compounds are released, enter our instrument, and then we can then separate them and obtain our chromatogram. And this was done for the air at a bus stop. And what we did, we measured the air for 1 hour to trap any volatiles because we wanted to see what we were exposed to at a bus stop. And this is an example of some of the things that we can see. So we see some aldehydes, we can see alkanes, we see methane, for example. But this sample, it's not that complicated. These are things that we were expecting to see at a bus stop. So for example, we see octane, which is a component of gasoline. Now we then went into the lab and also using the same analysis measured air in our lab, and we can see many more compounds. And part of that is because the lab is an enclosed space, whereas out at a bus stop, the compounds can diffuse, so the concentration isn't as high. But something that we found really interesting is that this was taken after COVID, and we see in particular peak number 5, isopropyl alcohol, which is actually due to sanitizers, and same with ethanol. So now moving on to the water story. In our lab, we are looking at measuring petroleum contamination in water samples. And this is something that is done commonly in environmental analysis labs in the industry where they have targeted analyses. So they're looking for a particular list of compounds. So I may have a sample that I bring to that lab. It's the sample that we can see is not very clean. It maybe has a smell, but based on their analyses, if they don't see any of the compounds on their list, they will say that the sample is clean. But again, from visual inspection, I can see that the sample is very dirty. I can smell something coming off of it. So we're trying to find methods that can actually measure the sample and can determine what that contamination is and try to broaden the methods that are available. So in general, the needs of the petroleum environmental analyses have surpassed the capacities of these industry labs, which have some techniques that are very, they're very easy to follow and they're cheap, but they aren't very good for analyzing so many new compounds that we're finding out about nowadays. So the goal is to improve and diversify analyses, meaning we want to look for a lot more while maintaining or making it cheaper. And some results from this, as we can see here, this is a one-dimensional chromatogram. So the FID is just a different type of detector, but we can see many peaks in these. And we also do see kind of a big mixture here where it's very noisy. So using the GCGC, we can actually separate out more compounds. So we see a lot of alkanes and compounds that are derived from alkanes that we wouldn't really be able to identify with one-dimensional GC, which is what is used in a lot of industry labs. Now, looking at environmental exposures, we can also use living organisms for these things. So we can measure both the metabolites from the organisms as well as any exposures. And we can use crab gill samples for this. So the gills act as filters for crabs who live both in water and can live in land. So we can look at the metabolites that are present in the gill samples that are produced by the crabs, as well as any kind of chemicals that can come from their environment. And the way that we do this, we obtain crab gill samples, which we freeze dry just to remove any moisture. We use some chemical solvents to extract the metabolites or the compounds that we're looking to analyze. And we also shake it just to really pull out those compounds. We then spin it down so that all of the solid material goes to the bottom, and then we can collect the liquid. But a lot of these compounds aren't really volatile, so they're not really gases. So we have to do a chemical reaction, which is known as derivatization. And this makes those compounds volatile so that we can then analyze them with our GC instrumentation, obtain our 2D chromatogram. And then after that, we have to process the data so that it can actually tell us what the individual compounds are. And we use some scripts which can actually tell us what the compound families are. So here we see the results from these crab gill analyses, which we see over 2,500 peaks, which are different metabolites or compounds present. And we see different compound families, for example, amino acids earlier on. We see some sterols like cholesterol, for example. We see fatty acids and sugars as well. But something that was interesting from these crab gill samples is that we also saw compounds from prescription medications and broad spectrum antibiotics just from the crab gill samples. So these could be used as a measure of environmental exposures. So it's possible that they may be exposed to these compounds from their environment. Moving on now into biological samples, we analyze biofluids as well as fecal samples, and these give us some insights into the human microbiome, and they're useful for providing information about a person's health. And to start off, I'll talk about the fecal project, and this is actually my main project. It's what I've been working on for the past 3 years, and what we are looking to study is the fecal metabolites, because they give us a direct link or a direct look into our gut microbiome. So how the bacteria that is present in our gut that helps us with digestion, how it's interacting with our health, and just the presence of those organisms. But feces and fecal samples are really difficult to work with just because they're not very uniform and they're very different. Sometimes they have less liquid, sometimes they have more. So we're trying to find a way to prepare the samples in a way that makes it uniform and easier to analyze. So I've used both a freeze-dried sample as well as just a frozen fecal sample. And we use some beads to homogenize it. So these metal beads were put into a tube with some of the sample. It was shaken at a very high speed, which really breaks up the material and also can break the cells in the sample, so it pulls out all of the intracellular metabolites. We then extract it with a solvent and again derivatize it, and then the following workflow is very similar as the crab gill samples. And here we see the chromatogram from the fecal samples, and we can see a lot of compounds. So we see amino acids, again, fatty acids, some sugars in here. Some of them weren't picked up, so these very intense peaks here are due to sugars. We can see an alcohol, and then we also see some sterols. And interestingly, we can also see whether a person has taken medication or has consumed anything. So for example, the donor of this sample is on medication and they had also consumed cannabis earlier on, and we can see the end products of cannabis metabolism in these samples. Now, we want to use biofluids as well to measure any environmental exposures. But the problem with this is that this derivatization technique makes it really lengthy to prepare these samples. It takes about 6 to 7 hours just to prepare 10 samples. And as we know, time is money. So if we want to use this for exposomics, it would just be really difficult when we have samples from many, many different people. So what we were wanting to do is just to eliminate this derivatization step here. And the way that we tried is using a different extraction solvent and drying a sample down with sodium sulfate. So this pulls out all of the water because we don't want to inject water into our instrument. It's not very good for the detector. So using this technique, it cuts our time down to less than 1 hour for just 10 samples. So that's very ideal. Now here we see the results from the derivatization technique, which is the one that takes 6 to 7 hours. We had a mega mix, so it's a mixture of more than 30 compounds that we know are exposure compounds. And using the derivatization technique, we can definitely see them and they are separated pretty well. And then we analyzed just a urine sample. So we do see that there are compounds around that same area. But if we mix the mega mix as well as the urine, we can still see those compounds and separate them out from the metabolites in the urine sample. And then moving on into the rapid extraction method, the one that cuts our time down to less than 1 hour, we can see that we still see these exposure chemicals in the mega mix, and they're actually even more intense. But the problem is that if we look at the urine sample, we actually lose a lot of metabolites. So it's not necessarily ideal, even though we still see them when we mix the urine and the mega mix. If we were looking to, as we do an untargeted analysis, we want to see anything and everything. So this technique is not the best. However, you may be wondering what this, we do see this peak here. You may be wondering what that is. And that's actually from caffeine. So if you were looking to measure caffeine in urine, this technique could work. And then more recently, we've been looking at breast milk samples and urine samples and trying to compare different methods for measuring these samples. So in this case, for the breast milk sample, it was all the same sample, same volume that was measured, and then same for the urine sample, it was all the same, but the difference was the different injection techniques. So similar to the blueberries, we did the SPME or solid phase micro extraction and the dynamic headspace. And then we also derivatized the samples just to compare them. And we do see a drastic difference. So with the derivatization, we see many more compounds compared to the SPME and the DHS. But again, the preparation technique that we choose it depends on what the question is and what we are looking to study. So in this case, if we were looking to study fatty acids, we may choose SPME over the derivatization method, because with this method, we lose a lot of the volatile compounds. But if we wanted to look, for example, at sugars, we would then choose the derivatization technique over SPME and DHS. But having these comparisons just allows us to know what exactly are the type of compounds that we can see with the different techniques, so that later on, if we have the different questions, we can choose the best option. And in the end, from all of these samples, we have to analyze the data so that we can come up with conclusions. But in our case, it's really, really difficult to do by hand because we have hundreds of samples with thousands of variables or individual peaks. And in some cases, for example, with the blueberry DHS, we had over 4,000 peaks. We can't just go through them manually and select the ones that are interesting. So we actually use different tools to help us with this. So we use chemometrics, machine learning, statistics, and bioinformatics, which are all tools that help us to apply statistical methods to analyze and evaluate our chemical data so that we can pick out the important features in the data. And ultimately, the impact of metabolomics research is that this will help us to complete the complex disease puzzle. Metabolomics and exposomics helps us to link all the different aspects of life. For example, our physical environment, our nutrition, any chemical exposures or our social environment, and using the different omics techniques, genomics, transcriptomics, metabolomics, and so on, can help us to then come up with better diagnostic tests, find prevention methods to prevent disease. It can then help us come up with questions for research and can make medicine better. I'd like to thank our different sources of funding. These are all how we obtain the funding to do all of the projects that I've talked about today, as well as our collaborators on the projects. And I'd like to thank my research group, the Harynuk Research Group, and Dr. Harynuk. And I would like to also thank all of you for listening to my talk, and I'd be happy to answer any questions.
Hi, it's Heather. I have a question. So did anybody taste the blueberries before you measured them? And was there a difference between, if you did it taste different than what the data showed?
So in our case, we didn't taste the blueberries just because we only got a very limited amount of sample. But when we were doing the preliminary studies, we were using just kind of blueberries that we bought from the store. And we tasted them, they tasted pretty good. And we can see a lot of like flavor compounds or, for example, terpenes that are compounds that are found in plants. We could see those compounds in the data.
Okay.
I had a question about the samples. How do you preserve samples if you collect things in the field and bring them to the lab and then want to get a metabolite profile from them? Is there a specific way?
So it depends on the sample. That's actually something like for fecal samples, that's something that's been pretty challenging just because fecal samples have so much going on. Like there's bacteria that is living. So if you collect the sample and it takes you a bit of time to bring it to the lab, the sample won't be the same from the moment you collect it to the time that we analyze it. And in that case, that's something that I'm also working on, is just trying to stabilize the sample. But for example, like air samples, that has been pretty, it's pretty standard where if you use the sorbent tube, there's the caps that you can put at the end, and it just ensures that the compounds don't escape during transport.
Can you like freeze them, or do you try to use a chemical to freeze them, like to prevent them from changing?
Yeah, so for these samples, like for example, with the blueberry samples, they are shipped to us on dry ice, or any of the fecal samples that I've analyzed from our collaborator, DNA Genotech, they send it to us on dry ice. And then when we get it, we store it at in a minus 80 degrees Celsius freezer. Other methods, well, at least like lab methods, is that if we freeze dry the sample, it can help with stability, just because it eliminates any of the moisture, which can, if there's like bacteria in there, it just kind of stops that bacterial activity because they kind of need water. But yeah, for the most part, freezing, or there's stabilization chemistries that are used.
Cool.
We have a question from Wayne. Is the intensity only relative to other components, or can it be used to estimate the quantity of a component?
So in our case, we do a lot of, it's just relative quantification, but you can do, you can actually quantify. So for that, we would have to know what exactly it is that we're looking for. So if I was looking for a specific amino acid, I would then do a calibration curve. So I would inject different samples with different concentrations that I know what the concentration is. And then when I inject my sample, I can then find where along the different concentrations it lies. And then that can tell us what the concentration of that compound is in the sample.
Okay. And another one from Roland. Wayne says, thank you. And then Roland asks, it seems your lab is doing many different unrelated projects. Is your lab a service provider, and are you approached by other groups?
Yeah, so in our lab, we do have some service contracts, and we also do collaborations just with other groups. So for example, the crab gills is a collaboration with the University of Antilles, and the algae samples are also from there. Whereas the blueberries, the ones that we're analyzing in the lab now, is a service project.
Okay. And Robert, you want to go ahead?
Yeah, I was wondering, feces is a really complex thing. Have you found any metabolites that relate to specific parasites, like worms or bacterial diseases?
Not necessarily. At the moment, since we're still in the early stages of the project, or in trying to find like the best methods for preparing the samples, we're mainly looking at how, like, if the homogenization method that we're using is really making the sample uniform. But we have seen, for example, the fatty acids, or short chain fatty acids. Those are compounds that are produced by bacteria in the gut. So when we use solid phase microextraction for those fecal samples, we could really see those compounds in the samples, and the different levels could be related to different bacteria.
Okay, and Roland, there's a reply from Carmen. If you check the chat. Are there any other questions? I guess I have one. Is there things that, how do I put this? Anyway, are there things that you can't identify?
Yes, so in our case, we have a mass spectrometer as our detector, which thankfully it can give us like a tentative or tentative identification of the compound. So it matches what the signal that we have from the compound in our sample to different libraries of known compounds. But in some cases, there are compounds that that signal doesn't match up. So they show up as unknowns in our data. And in order to really find out what it is, like we can look at again, like in libraries, try to see if we can find something that matches. And then in the end, we would have to then purchase that that compound, inject it, and see if it comes out at that same time.
Okay, thanks.
No worries.
Okay, so if we have no more questions, I'd like to thank Kieran once again and for teaching me how to pronounce metabolomics. And thank you all for joining us.
Thank you so much for having me.
It was very interesting. Thank you.
All right.
Thank you
Thanks,
so much.
Karen. That was fun.
Yes.
Complicated.
Yeah.